Okay, so let's go back and review the genetics. When we talk about CML, it actually is a chromosomal rearrangement. That means a little bit of chromosome 9 and a little bit of 22 are actually translocating. They are swapping with each other. You can see how the ABL gene and the BCR gene, these are the two genes that are essentially related to this disease. These two genes are normally existing on two entirely isolated chromosomes. However, due to this translocation, you can see that the ABL gene and the BCR gene are now on top of each other. And you end up with this Philadelphia chromosome, this chromosome 22, that's essentially a fusion between the two chromosomes. Now, we've talked a lot previously about how a genotype determines phenotype, and here we can actually see this again. You can kind of reiterate your genetics. The genotype, the change in the DNA, this DNA mutation that occurs due to the simple switching of this DNA, so this sequence has been moved, and the sequence is going to actually produce a mutation that's going to lead to an altered phenotype. And this altered phenotype is due to a protein that's going to cause the disease. So the two genes that are responsible for causing the CML, it's ABL1 and the BCR. ABL1 actually stands for Abelson-Murine Leukemia Viral Oncogene Homology 1. How's that for a mouthful? We'll just call it ABL1. So this is actually a tyrosine kinase. Now remember, kinases are enzymes that transfer a phosphate group from an ATP to a protein. You're actually going to change the shape of that protein, so post-translational modification. More on that in a second. ABL1 gene plays a role in cell differentiation, cell division, cell adhesion, and stress response. So normally this gene plays a role in regulating all these. The BCR gene stands for breakpoint cluster region. And actually this is kind of interesting. We certainly know what this gene does when it's fused to ABL1, but the normal expression of the BCR gene is actually not really well understood. But the two of these together, the fusion of the BCR to the ABL, produces a protein that has the tyrosine kinase activity from the ABL1 and the expression of the BCL gene. And this fusion protein is actually going to have an increased tyrosine kinase activity. So essentially what that means is this protein is going to have no off switch. Normally the ABL1 gene is regulated. It's going to be turned on at certain times, it's going to be turned off at certain times. But when it's coupled to the BCR gene, this little bugger never turns off. So it has no off switch. And that's what leads to this signal transduction cascade, the enhanced proliferation, adherence, and apoptosis inhibition. So remember, just to review, post-translational modifications can include glycosylation. So essentially what happens here is a carbohydrate chain is going to be added to your protein. It can include cleavage, so the protein itself could be trimmed, cut and trimmed. That would include a signal sequence. Maybe it reached its destination and that signal peptide needs to be removed. It could also be for insulin. A whole set of amino acids in the polypeptide are actually removed in insulin. It could have chemical modifications. It could have addition of functional groups or proteolysis, right, targeted for degradation. These are all post-translational modifications that can occur. Okay, so the mutant protein produced by the BCR-ABL protein actually plays right into this. Remember, what it's doing is it's adding a phosphate group to proteins. So it's adding an additional functional group. And the proteins that it's modifying are going to be changed. Their shape, their tertiary structure is going to be altered, and their function is going to change. So a TP is going to have one of its phosphate groups removed. It's going to be an ADP at the other end, and that phosphate is actually going to be added to the protein, whatever that kinase is doing. So remember, the tyrosine kinase, the BCR-ABL fusion protein, is acting as this kinase. So essentially, what this fusion protein is doing is adding phosphates to everyone. The thing really just can't control itself. So because the fusion protein is now on, right, it's constitutively on, the normal proteins that it's interacting with can actually become hyperactive. It's like you have a friend who just keeps talking in your ear and finally you're like, I can't take it anymore. That's what this ABL-BCR fusion protein is doing. It's causing other proteins in the area that it's interacting with, that it's adding that phosphate functional group to be hyperactive, to change their behavior. So interestingly, something I want to stress is that the central dogma actually is maintained with cancer cells, with the CML. The central dogma is that DNA is going to be replicated and then that DNA is going to be transcribed into messenger RNA. That messenger RNA is going to be translated into protein. But the interesting thing is that the mechanism of the central dogma of gene expression is actually not changed with this fusion protein. The DNA is altered. Remember, we have those two chromosomes where they're translocating, so the DNA is actually being swapped out. So there is a mutation that's occurring. The DNA is being altered, but like a good cell, that mutant DNA is being transcribed faithfully. The messenger RNA, which is different than the wild type one, than the normal one would expect it to be, is going to be translated faithfully, and you're going to end up with a polypeptide that is then different. So the protein is going to have a new function. So everything works, it's just that the blueprints are different. As for example, you got blueprints for a car. If the blueprints were mistaken, you would end up with a car that actually looks altered as well, right? The BCR and the ABL1 fuse together. That car right there would be the result of a fusion protein between two parts of two different genes. So instead of having a normal blueprint where you end up with two cars, you end up with that fusion protein. So just a really brief view on the mechanics. Remember, transcription and translation, both of them occur in three phases. You have initiation, elongation, and termination. Now let me say that transcription, elongation, and transcription termination are actually not altered at all. So remember from our diagram, you can see that the DNA template strand is being replicated or copied faithfully when it's being transcribed. The growing RNA strand is going to add a U to the A, and you can see that the phosphodiester bond is going to be catalyzed between a 3' hydroxyl group from the previous nucleotide and the phosphate of the nucleotide being added. Sure enough, there's going to be a CDCTP being added to the next nucleotide based off of complementarity to that existing template G. And in fact, when you get to termination, we don't really understand the exact mechanism of eukaryotic termination, but the process is going to function the same way. Transcription will be terminated, and you're going to end up with your pre-mRNA that's then going to be processed. Also, translation is going to function the same way. The big difference, the one that we're going to be stressing mostly in this review, is transcription initiation. So let me spend a little bit of time talking about initiation. Initiation requires a promoter, a special sequence of nucleotides that act as a bat signal and a landing strip for the RNA polymerase to come, land, recognize that there's a gene there, and initiate transcription. So the interesting thing about the CML protein, this fusion protein, is that, remember, we have the ABL structural gene translocated and transplanted behind the BCR promoter sequence. So think about the implications of that. The bat signal is essentially going to be saying, here's a BCR protein, but when the RNA polymerase comes and transcribes the gene based off of that bat signal, it's actually going to be producing the wrong protein or a different protein. So the image you're looking at here is a prokaryote. The reason I know that is because you can see the sigma factor binding to the promoter region. But the principle is going to be the same between our eukaryote and our prokaryote right here, in that proteins are going to be binding to the promoter sequence to initiate transcription. Now, the thing about the BCR is it's considered an oncogene. And the reason is because BCR is what we call constitutively expressed. That gene is always produced. The ABL gene, normally, is going to be produced at sometimes and not produced at sometimes. The implications of this, when you put the ABL structural genes behind the BCR promoter, is that, well, the ABL genes are always going to be expressed. In the little figure down there, pretend like you're taking the wild type ABL protein, that tyrosine kinase, and you have it behind its normal promoter. You'd expect it to be transcribed and translated when directed by the cell. Now this is going to be induced. Sometimes it'll be expressed, sometimes it will not be. But what happens when you put ABL behind the BCR promoter, you get an overexpression of that protein. Now again, you can see the implications of this. Remember, this guy is a kinase. It's going to be adding those phosphate groups to protein, and it's going to be overproduced. It's going to be cranked out constantly because this BCR promoter is essentially always on. So this poor cell essentially has a bat signal saying, you need to land here and constantly transcribe and therefore translate this gene, but the gene behind it is something that shouldn't be done that way. It should be regulated. All right, so let me take a step back and talk about kind of the mechanism on how the difference between a BCR and an ABL promoters work. And this has to do with the eukaryotic gene regulation. There are two basic requirements for transcription. First is access. the RNA polymerase needs to be able to have access to the promoter region. Access can be denied due to the binding of DNA to what's called the histone proteins. So you can see here the histone proteins are these green balls and the DNA is wrapped around it. Now the DNA plus these histones is called chromatin, right? Inside our DNA, you've heard this term before, we have heterochromatin and euchromatin. So this plays an actual very important role in gene regulation in eukaryotes. Heterochromatin is going to look like these two examples. You can see how the DNA is very tightly wound around the histones, and therefore those genes are not going to be easily accessed by the transcription factors and by the RNA polymerase. Compare that to the DNA that's opened up, euchromatin. The DNA here is opened, accessible. You can see the RNA polymerase actually landed right in there inside the promoter site, and now you're going to have active transcription. So those genes are going to be turned on. So remember, inside the cell, when we talked about differentiation, you have the Christmas ornaments, the DNA that's all packed up. Well, that's going to be the heterochromatin, and the DNA is going to be tightly wound around the histones. The cell that's using a particular set of genes are going to be opened up. The Christmas ornaments are going to be opened in December, and the cell is going to be using those genes, actively transcribing those genes. So differentiation of a cell is going to determine what's going to be opened and what's going to be closed. So first is access, a significant way that eukaryotes regulate gene expression. The second way is actually simply the transcription factors themselves. A plethora of transcription factors able to recruit RNA polymerase and access of those guys to the promoter site are actually going to regulate transcription. It's going to allow transcription to occur. Now, the nasty thing about this ABL-BCR fusion protein is that it's actually going to influence both. It's going to influence the access, the heterochromatin versus euchromatin formation inside a cell, and also the amount of transcription factors that are going to be produced. So as we saw earlier, some of the effects of the fusion protein are to enhance proliferation, reduce adherence, and inhibit apoptosis. So you can think about the cell cycle. We have RB, retinoblastoma, inhibiting the cell cycle. Well, if the fusion protein, the CML fusion protein, inhibits RB formation, that cell is no longer going to be arrested at the cell cycle checkpoint. It's going to be able to go through too fast. So in this way, the cell, by simply changing the access of the promoters to transcription factors, can influence what's going on inside the cell. Okay, so remember epigenetics. This was a topic you guys tended to find pretty interesting. Remember, this is actually a change in the phenotype of an organism that is not due to a change in the DNA sequence. The change in the DNA sequence would be considered Mendelian inheritance, right? A big A versus a little a. But what happens in epigenetics is that you're actually changing the rate of gene expression. So again, the fusion protein is not necessarily altering the DNA sequence itself. In fact, it will, once a person's cancer progresses, it actually, when the cell goes past the cell cycle too quickly, you can actually have an increase in the chromosome number, which can create some serious issues. But the fusion protein itself directly is not causing a change in the DNA. What it is doing is it's causing a change in the expression of various genes. So these are kind of some interesting little side-by-side pictures of identical twins, and you can actually see the role that the environment is playing in changing the phenotype of these individuals. You can see the younger kids tend to look much more similar. The woman in the middle, you can see a little difference between her and her identical twin. And then the older you get, the more epigenetic modifications that are occurring. They have the same genes, they have the same Mendelian inheritance, but they have a markedly different phenotype, right? This poor guy is bald and like 4 inches shorter than his brother. So somehow he got the short end of the stick on those epigenetic modifications, but he actually looks happier, doesn't he? Maybe I should shave my head. But the same thing is happening with these cancer cells, right? The cancer cells, besides that mutation that's occurred due to the Philadelphia chromosome, but these cancer cells are going to have the same DNA. So one of the other key concepts in the peer-reviewed paper was talking about transcription factors, and I've mentioned this a couple times, but I do briefly want to kind of give a little review, and again related to the CML. The most common consensus sequence is actually what we call the TATA box. You can actually see down here the sequence, a TATAAA. Consensus sequence, it just means that it's found repeatedly in different organisms. That sequence will be where the RNA polymerase is directed to land, and then you can see the +1 is going to be the start of the first exon. There's also the CAT box and the GC rich box. These are, again, are consensus sequences that proteins are going to bind to. And now these proteins are actually the transcription factors. So I always think of it as herding cats. The RNA polymerase is just going to diffuse randomly around through the nucleus. And it's not going to know where one gene is versus the next versus uncoded regions of DNA. Now it's going to actually be brought to DNA by these particular transcription factors. Remember, the RNA polymerase is agnostic. It doesn't care what gene it's actually transcribing. So the transcription factors are crucial for bringing and directing the RNA polymerase to the site. And it does this through binding of the tau box. So you can see these general transcription factors include TF2D and various other ones that are going to bind to this promoter sequence. And you can see how the TF2D is going to bind first, and then TF2F is actually going to recruit and bring the RNA polymerase. And then there's going to be a couple other general transcription factors. Once all of these general transcription factors are bound to the promoter, you can see that the RNA polymerase will finally be released and begin transcribing the gene. So when we say the general transcription factors, we're talking about the basic required proteins to bind to the promoter site to allow transcription initiation. How quickly these proteins bind are going to influence the efficiency and are going to influence the rate of transcription. So both of these, the accessibility and the amount of transcription factors, both of these play a role and play into signal transduction, or what we call coordinated control of gene expression. And here's an example. You can see how all these proteins are playing a role in directing transcription of a particular gene within a nucleus. And again, to get back to cancer and CML, each tissue, each specific tissue is going to have its own signal transduction cascade going on to produce the genes necessary. The muscle cells are going to have all the genes coordinated to function as a fully functional muscle cell. And the intestinal cells are going to turn on and off genes to allow it to function as an intestinal cell. So each specific differentiated specialized tissue type is going to have genes regulated based off of the accessibility of the DNA versus the heterochromatin, euchromatin, and the binding of these transcription factors. So these transcription factors play a huge pivotal role in regulating transcription. So they need to be tightly regulated. And what happens is the transcription factors that a cell produces are going to affect the cell's activity. If a cell wants to change its developmental fate, if it wants to change its metabolic pathways or change its metabolism, well, it'll simply induce a different set of transcription factors. And all those proteins will then bind, recruit, and cause transcription to occur for a different set of genes. So this is the part where that fusion protein, the BCR-ABL fusion protein, really plays a significant role. The signal transduction cascade in a cell that has this fusion protein is going to be short-circuited. Now the reason is because normally you have signal molecules being received by a cell. And you can see how these signal molecules are going to bind and cause a change in these receptor proteins. Inside the cell, you're going to have proteins activated, and then it's going to lead to a cellular response. So the picture on the right, you can see again how a growth factor bound to the receptor is going to cause a whole series, a whole cascade of events where inactive proteins are going to be activated by a phosphate group being added to it. And notice how this cascade of events is leading to activation of a gene inside a nucleus. So remember, our BCL-ABL fusion protein, this little sucker is stuck on. It cannot turn itself off. So it's going to cause a dysregulation on all the events that are occurring in several signal transduction cascades. Because it's always on, notice how the ATP is constantly having that phosphate group removed, and the substrate that it's acting on is going to be changed. So whoa, before you get too overwhelmed, what you're looking at here is the actual signal transduction cascade that occurs with the BCR-ABL fusion protein. So again, this is the cytoplasm of the cell, and the growth factors are normally going to be received. That's going to tell the cell, hey, it's time to proliferate. It's time to induce a cascade of genes that are going to cause proliferation. Now notice what happens here is the BCR-ABL fusion protein is actually short-circuiting this. So instead of the cell needing the growth factor from the external environment, the BCR-ABL is able to do it by itself. So because it can no longer be turned off, because it's always on, it's going to be adding a phosphate group and changing the shape of the downstream proteins. You can see GAB2 and RAS, and then that's going to cause Even if RAS and RAF and NEC and ERK1 and 2, even if these guys are completely normal, because the protein, because that fusion protein upstream has short-circuited this whole effect, you can see that it doesn't matter. The cascade is going to be set off. It's like dominoes. Once you push that first domino over, even if it was pushed over at an improper time, you're going to get these downstream effects and you're going to get gene transcription. So in this case, we're looking at the cascade that's signal transduction cascade that's affecting growth. Incidentally, the GAB2, you can see it's going over to the mitochondria, and all those skull and crossbones have to do with apoptosis. So the mitochondria actually play a role in programmed cell death within our cells, and notice how a lot of these skull and crossbones are being crossed off. And the reason is because the BCR-ABL1 is actually, again, causing an inhibition of apoptotic genes. So again, to take a minute to review in our review, this fusion protein, it induces proliferation, it reduces apoptosis, and it decreases adherence. And I think I've said this before, adherence just has to do with the ability of a cell to stick to its neighbor. And a decrease in adherence means it's no longer going to be wanting to stick to its neighbor, and it's going to float around, and that's what we call a metastatic cancer. Here are your TF2D. They're bound to the promoter site. And notice how the green blob here is actually the RNA polymerase. It's being recruited to the transcription start site by these transcription factors. They're bringing in this RNA polymerase to the promoter region. And what will happen with this fusion protein is that there's going to be more of these transcription factors. They're going to be around in a higher number, and this whole signal transduction is going to be short-circuited, and you're going to get more transcription of those proliferation genes that are going to cause those cells to grow out of control. And once this messenger RNA is produced, it's going to be sent out into the cytoplasm, and this gene could be totally normal. But because it's being dysregulated by that fusion protein, the cell is going to act funny. Well, now that I have everybody completely depressed and scared, let me say that CML is one of the most treatable cancers that we know. In the next part, we're going to talk about the treatments that we have for CML, and then following that, we're going to talk about how these cancer cells are becoming resistant to those treatments.